Okay, so, recording. All right, so we talked last time about post-transcriptional modification, RNA modification, basically. That pre-mRNA has to get modified. So it gets a guanosine cap, and it gets a polyadenylated tail, so a bunch of adenosines added on the end, so there's an actual tail, and it also gets spliced. So what gets spliced out? Remember I said there's different types of sequences that are in DNA and mRNA when it's first made. Which pieces get cut out? The introns get clipped out, right? So all the introns get clipped out, the exons are remaining, and they get glued or spliced back together. So those are the genetically coding parts of the transcript. So they get spliced, capped, tailed. Why does all this happen in eukaryotes? Why do we have this RNA processing or post-transcriptional modification in eukaryotes?
Prokaryotes have both transcription and translation together.
That is true, and prokaryotes is more simple, right? They're transcription and translation is all happening at the same time. It's not parceled out, right? It's all just happening right in sequence. But what does this help do in eukaryotes? What does all of this help do? The cap, the tail, the splicing, sending it out. Do you remember?
It makes it more stable.
More stable, yes. More stable so it'll last longer, so that it can be exported out the nucleus. It can make it out to the cytoplasm. And then the ribosomes and rRNA and all of that can come on and translate it into protein. So it allows it to be more stable, resistant to hydrolytic enzymes that would break it down and chomp it up. It's single-stranded nucleotide, right? So it'll be broken down. So this really helps to allow its longevity, make it more stable, more resistant to being broken down, so it can last for longer. So this can be shipped out, last for longer, and actually be translated into protein. So it helps to facilitate it out of the pores, out of the nucleus, and get it out to the cytoplasm, protects it from hydrolytic enzymes, increases how long it's gonna live, so that in the complex eukaryotic cell, it can make its way out of the nucleus, get to the ER or to the free ribosomes, and actually get all the assembly for making the protein, and be able to have time to do all that. Oops. Helps the ribosomes also attach to the prime end. And that's going to be needed in the next step that we're going to look at in translation. And going from transcript or mRNA to protein. Okay, so the splicing, there's a whole set of enzymes and proteins and machinery that actually come together we call the spliceosome. And the spliceosome is this big machinery, it's like the fruit ninja, that basically chops it up and puts back together the exons, removes the introns, and puts the exons together. So in eukaryotes, the exons and introns are in the genes. So they have to be sort of deciphered and put back together the right way. Because remember, it's kind of counterintuitive, but the exons are the ones that code for the genetic sequences that are important. The introns are material that needs to be removed. They don't code for the actual protein. So exon sequences that are translated into actual protein, intron sequences that are not translated, we call them junk DNA, but really that's a misnomer, because sometimes there's important information that are in the actual introns. They have regulatory sequences. They're involved in sometimes the binding of different proteins and involved in regulation of gene expression. But they're not per se in the actual what's going to be translated into protein. Okay, so the introns have to be cleaved out. So for translation, the exons need to be brought together, the introns need to be removed or clipped out so that the RNA can actually have its final mRNA form. Okay, so that's why I did the little fruit ninja, because we call this RNA splicing, and it's another part of this modification. The mRNA gets a cap, gets a tail typically, and then it gets spliced, okay? So it's showing here there are intron sequences that need to be removed. These are the actual exons, these three white pieces in this particular transcriptional unit or this gene. These are the pieces that code for the actual protein. So the importance of this gene. So these need to be actually removed. And then these need to be brought together. This one. Okay. And this is an example of a beta globin gene. This is actually one of the subunits of hemoglobin. So remember we talked about hemoglobin being a quaternary protein. It's made of 4 separate globular proteins. Those each come from separate genes. They're actually transcribed and translated from 4 separate genes, and then ultimately covalently bonded together. Okay, so RNA splicing, that's what we have to do, and that's why I say it's like fruit ninja. You're basically, the right enzymes come on and cleave apart at these recognition sequences, and then bring them together. So this is all happening where we call DNA to pre-mRNA, and then the pre-mRNA and then it hasn't been modified. Once it's modified, now you have a full-fledged RNA, typically has a cap and a tail, and it leaves so it can actually undergo translation out of the cytosol. Here's an example, pre-mRNA. The final splicing that happens ends up bringing together those three these three exons in this particular gene, and so they're one continuous sequence with a tail and a cap. And then there are these little regions right at the end. So in the end of this gene or this transcriptional unit, there are these UTRs. There's a 5' UTR and a 3' UTR. This is just this end little region right here, right downstream of the cap and right upstream of the poly A tail. Those we call untranslated regions. UTR is an untranslated region. Your book talks all about these and what all of these things mean. But the 5' and 3' UTRs, those are not going to be translated into protein, but they're part of this transcriptional unit. Okay, so they're included in the first and the last exon. Now note, by the way, there can be lots of exons in a gene. I opened with a gene showing you one of the contig sequences of a gene, and it's all in these separate sort of pieces. And there can be hundreds, dozens of exons, or there can be just a couple, depending on how big that protein is, or how big the gene, respectively, is. All right, so we call these spliceosomes that do this RNA splicing. Not always, but usually RNA splicing is carried out by these big factory spliceosomes. So these spliceosomes, there's a whole bunch of proteins, several small ribonucleoproteins. So this is a different type of basically a RNA protein in snRNP. Small nuclear ribonucleoproteins, so these are RNA with protein. And they recognize these splice sites. So the given sites between exon and intron, those boundaries, they can recognize those places and cleave. So the RNAs of these spliceosomes also actually catalyze the splicing reaction, the actual cutting reaction. So these special small nuclear ribonucleoproteins are actually RNA and protein, but they have an enzyme function. So they're actually able to cut. They catalyze a reaction. Okay, so it's just a little unique because most enzymes are proteins. These aren't really enzymes, these are a mix of sort of RNA protein. And they're really small little pieces. But they all come together to make up this spliceosome. Okay? So you have the RNA pre-mRNA transcript. In this case, it's just showing two exons and one intron right in the middle.